Hello, everybody. This is Dr. Karen's, and this is a short lecture related to the genetics chapter in our microbiology course, which is chapter 7, and it's designed to help you work through that genetics activity that I've assigned. So let's start with talking about DNA replication that is related to this activity. So the purpose of DNA replication. Remember, we're trying to make a copy of our DNA. Why do we want to do this? Because we need it for cell division. When that cell divides, it needs to be able, daughter cells need to be able to have the right number of chromosomes. So we have to replicate the DNA first, and then the cell can divide. And because cell division happens only once during the life of the cell, so does DNA replication. And in prokaryotes, this is going to happen in the nucleoid region of the cell because that's where the DNA is. In this process, we start with one double-stranded DNA molecule, and we end with two identical double-stranded DNA molecules. And we need to do some base pairing rules here because we're dealing with nucleotides. And these rules for DNA replication, as you should know, A goes with T, G goes with C. When we say that DNA replication is a semi-conservative process, this means that each of those daughter DNA molecules that are made during DNA replication each consist of half old DNA and half new DNA. The enzymes to make a note of for this process, DNA helicase, which unwinds the double strand, DNA polymerase adds new nucleotides to the growing chains, and then DNA ligase will fill in the gaps. So this is an image that you've seen in chapter 7 of the textbook showing you a circular bacterial chromosome, and then as it's double-stranded DNA, and as a result of replication, you get two double-stranded DNA molecules in the form of these circular chromosomes. It's hard to tell from this image, though, that they're identical because you can't see the sequences. So what does this look like if we use actual sequences? So here is a double-stranded DNA molecule. It consists of two strands that are complementary to one another. You can see the A goes with the T goes with the A, so on and so forth. We can denote these complementary strands with a positive and negative sign. So there's a positive strand here, and there's a negative strand. Positive and negative strands are complementary. You cannot have complementary positive strands or complementary negative strands. You have to have one of each. During DNA replication, that DNA helicase enzyme has to separate the two strands, and both of these strands will be used as a template to build new double strands. DNA polymerase is going to use those templates, which are shown in black, to figure out which new nucleotides to put in place so we get double strands as a result. And it uses the base pairing rule. So if there's an A on the parent strand, then we put a T here. The A goes with the T goes with the A, the G goes with the C, so on and so forth. And then the DNA polymerase will do the same thing with the other template. So as a result, what we have are two double-stranded DNA molecules. And if you look at the sequences, they are both identical to one another. They are also both half old and half new. And if you look at these sequences, they are both identical to what we started with. So we basically just made a copy of what we started with. So if I were to give you a sequence of just one strand of DNA and ask you, what is the complement? You just need to follow those base pairing rules. If I were to give you a double-stranded DNA molecule and ask you what is the result of DNA replication, you know you'd have to write out two of those double-stranded DNA molecules. All right, so that's DNA replication, but let's move on to what's called the central dogma of genetics. This is also part of that genetics activity. And overall, this is talking about the process by which we go from gene on a DNA molecule to a protein. That's the whole point of having genes, is they all code for different proteins. The proteins are what does all the cellular work. There are two steps to this overall process. First, we do RNA transcription. And in a nutshell, what does that mean? It means we're taking the message on the DNA and converting it into a messenger RNA molecule. And when we're talking about this central dogma, meaning going from gene to protein, when we talk about transcription, we are specifically talking about making messenger RNA. But note that there are other types of RNA, and anytime a cell needs to make any type of RNA, it's going to do transcription to do it. But in this case, we're talking about making messenger RNA. The second step involves RNA translation, which is taking the message that we just put on the messenger RNA, which is shown down here, and converting that into an amino acid sequence, which will become our polypeptide, which becomes our protein. All right, so let's look at transcription in a little more detail. So the purpose of transcription in this case, since we're talking about central dogma and going from gene to protein, we are talking about the first step in protein synthesis. We want to copy that information that's on that DNA gene and put it into a messenger RNA molecule. This can happen many times during the life of the cell, and it's going to happen in the nucleoid region because that's where the DNA is. We start with one double-stranded DNA molecule, and we end up with one single-stranded messenger RNA molecule. Because we're making RNA here, the base pairing rules are slightly different because RNA doesn't have any T's in it. It has U or uracil instead. So the base pairing rules now are A goes with U, G goes with C. And the enzyme that's important in this process is RNA polymerase, and that is what is going to be reading the DNA template and determining which RNA nucleotides to add to the chain. So we've seen this picture here. This is in the textbook. It's showing you our double-stranded DNA molecule here. In the brackets is what is the actual gene, and that's the information that needs to get transcribed. RNA polymerase is going to read one of those DNA templates and use that to put together the messenger RNA. But what does this look like with using sequences? So let's go back to our DNA molecule here. This is the one we saw earlier, our double-stranded DNA molecule. This is the gene. The gene, more specifically, is on the positive strand of the DNA. So temporarily, that double-stranded DNA has to separate. And only one of these strands now is going to be used as our template. Which strand? And the answer is the negative strand. And why is that? You will see in a second. So that negative strand of DNA is what the RNA polymerase is going to read and use base pairing rules to put together the messenger RNA. So you can see the T on the DNA, well, that means an A for the messenger RNA. A on the DNA, that means a U on the messenger RNA. There's no T's in RNA. So on and so forth. And what we have here temporarily during this process is a double-stranded DNA-RNA hybrid until the RNA polymerase reaches the end of the gene and then transcription stops. And we're left with our single-stranded messenger RNA. That DNA template just goes back to its partner and becomes a double-stranded DNA molecule again. So I told you that the template that is going to be used during transcription is the negative strand of DNA. And the reason for this is because messenger RNA has to be positive. And why does it have to be positive? Well, I told you earlier than that the gene that you find on the DNA is on the positive strand. And this whole process, we're trying to get the information from the gene into a messenger RNA. So if the gene is on the positive strand of DNA, then the messenger RNA also has to be positive. And if messenger RNA is positive, that means the template that you use to make it has to be negative. So if I were to give you a double-stranded DNA molecule like this, and then ask you to tell me what the messenger RNA sequence is, you have to remember, okay, I'm doing transcription. I'm only going to use one strand of my DNA as a template, and that template has to be negative because I know that messenger RNA is positive. Look at that sequence of our messenger RNA. It's exactly the same as that positive strand of DNA, except for the T's. There's U's instead. It's the same information. All right, so we have our DNA gene information now embedded within this messenger RNA molecule. But what is the next step? Translation. We're not done. We need to turn that message into an amino acid sequence, which is the purpose of translation. It's the second step in protein synthesis. And we're trying to translate that message on the messenger RNA into an amino acid sequence, which will be our polypeptide, which will be our protein. This will also happen many times during the life of the cell, and it's going to happen in the cytosol at a ribosome. This happens only one messenger RNA molecule is needed as our template, and the end result is a polypeptide or an amino acid sequence. We're dealing with RNA again here, so our base pairing rules are the same as they were in transcription. A goes with U, G goes with C. And ultimately what's happening, and I'm going to show you some pictures in a minute, but what's going on here? The ribosome is reading the message on the messenger RNA, and it's bringing tRNAs to base pair with the messenger RNA. And it's doing that 3 nucleotides at a time. So the messenger RNA contains codons. Every 3 nucleotides is a codon on the messenger RNA. The tRNAs also have a special triplet sequence called an anticodon. The anticodon and the codons base paired together. They are complementary to one another. So we're base pairing tRNAs with messenger RNAs. Why is that important? Well, because the tRNAs, which are shown here, are carrying amino acids, and that's what we're ultimately trying to do. So if the ribosome is reading the messenger RNA, codon by codon, and bringing complementary tRNAs to base pair with those codons on the messenger RNA, when that's happening, those tRNAs are bringing with them amino acids. And so ultimately, it's the codon sequence on the messenger RNA that determines which tRNAs can base pair with the messenger RNA, and therefore which amino acids get strung together. Here's another picture from the book, and it's showing you what happens when you add more tRNAs. But basically, the ribosome reads the messenger RNA 1 codon at a time. Complementary tRNAs with their anticodons will base pair with the codons on the messenger RNA and bring amino acids with them. As that's happening, the amino acids get chained together. When the ribosome gets to the last codon, that is the stop codon, and the whole process ends, and what we're left with is a chain of amino acids. But what does this look like with sequences? Here's our messenger RNA sequence that we made during transcription. This is our template. And what happens? Like I said, the ribosome is going to read that messenger RNA 1 codon at a time and bring a complementary tRNA to base pair with it. So you can see the AUG, the codon here on the messenger RNA, is complementary to the UAC anticodon on the tRNA. The tRNA brings an amino acid with it. The next tRNA, complementary to the next codon on the messenger RNA, and it brings its amino acid with it. So we have a temporary tRNA, mRNA double-stranded molecule here that's only exists during the process of translation. So if you're given a messenger RNA sequence, you can predict the tRNA sequence because it's complementary to the messenger RNA sequence. This last codon in this example, there's no tRNA for it because it's a stop codon, and that tells the ribosome to stop the translation process. So in this situation, we are left with a 2 amino acid sequence protein. In reality, proteins are a lot longer than that. This is just to simplify it. How do you know that this first tRNA is carrying this methionine amino acid and that this second tRNA is carrying this alanine amino acid? How do you know that? It's determined by the codons on the messenger RNA. I cannot stress that enough. It's the codons on the messenger RNA that determine which amino acids are going to be put together. There is a chart in Chapter 7 of our textbook that shows you which amino acids correspond to which codons on the messenger RNA. So you could be given a messenger RNA sequence, and I could ask you, what's the amino acid sequence from this messenger RNA sequence? And what do? Well, you look at your codons on your messenger RNA, and you go to that chart, and you find that codon, and it'll tell you which amino acid corresponds to it. And that's how you figure that out. So this is the end. So what did we do? What did we do? We went from our double-stranded DNA molecule, we made a messenger RNA molecule during transcription, and we made an amino acid after translation, amino acid sequence after translation. So we basically have gone from gene to protein here. The rest of this activity deals with mutations. And I'm not going to go into detail, but I just want to point out that the 2 questions ask you to do 2 different things. So number 6 asks you to insert a T in the 6th nucleotide position. So insertion is kind of like, I'm looking up, in the figure in the book, that's kind of like a nonsense mutation. Oh, I'm sorry, no, it's not. It's a frame shift mutation. I don't know why I said that. So inserting means adding, really. You are adding a nucleotide. And if you're adding it into the 6th position, if you're adding it into the 6th position, that means everything that was in the 6th position and onward gets shifted. Whereas in number 7, it's asking you to substitute a T in the 7th nucleotide position. That means swap out. Whatever was in that 7th position, put a T in there instead. So there's a difference between inserting and substituting. In both of these questions, they're telling you to do something with the DNA, but then asking you, well, what is the new amino acid sequence? So that means you have to go through the steps of transcription and translation to figure out what those changes in the DNA are going to do to that amino acid sequence. So go back and look at what you had to do before on that worksheet, you have to do again here. All right, so that's it. I recommend, if you didn't have that worksheet out already, go back, watch this again with the worksheet handy. Pause where you need to fill out that worksheet. Converse with your lab mates, converse with your group members, and then you'll have opportunity to ask me questions.